Welcome, everyone. Pleasure it is to kick off the seminar series called 2023 with international colleagues coming all the way from Finland. So sometimes we write a paper and nobody in the world reads it except our students that we force to read. Sometimes you write a paper and it just takes one person to read it from across the world who has similar interests and by effect, I guess, and grants that are written, not funded, and then finally funded. We get guests from Finland who come to us from the University of Jyväskylä, which is a really goodness. They're so humble. I've had several meals with them and I never knew the distinction of this place to study exercise physiology or health sciences. I think they are highly ranked as far as world institutions. Their acceptance rate is 7% of the students that apply there get in. So anyways, it's a great place. We are so lucky to have them. So welcome. And I'll just go ahead and introduce them both now because they're going to give two little short talks. It'll be a little bit of a unique. So I'm really excited to see their face. We have Enni Hietavala, who is a lecturer in biology of plant physics. Exercise in the context of menopause and athletic population. In her PhD project, she studied the effects of dietary acid load and exercise performance in different age groups. Currently, her main focus as a lecturer is in developing a new program in sport of science coaching. Hietavala is a member of the estrogenic regulation of muscle apoptosis, estrogen, microRNAs, and the risk of metabolic dysfunction research group. Has received funding for her own sub-study investigating the relations between mental well-being and health during menopause. In addition, she works as a sports nutritionist, especially with professional ice hockey players, which we have Sira Karvinen, Academy Research Fellow, fascinated to discover the molecular, cellular, and physiological mechanisms underlying healthy aging and longevity. Her key interests include investigating how physical activity, genetic background, and hormones affect biological aging. Effective genetic background on lifespan, which prompted her interest in the molecular mechanisms that underlie health and longevity. A 3-year postdoctoral research project, which focused on microRNA signaling, extracellular vesicles, and HDL particles in response to an acute bout of exercise. A new 4-year project concentrating on exercise and cellular senescence. During her career, Karvinen has obtained several awards and grants, podcasts, which I was on, both from national and international sources, has published 24 peer-reviewed articles in high-impact journals such as Cell Metabolism and Journal of Exercise. Without further ado, please join me in, might I say, a warm Missouri welcome to our Finnish guests. Thank you so much.
Thank you so much for having us, Vicky, and thank you for the kind introduction. I think Enni will pass some. We brought some sweets, even though this isn't nutrition, I know, but you know, maybe one candy here and there doesn't hurt. So please. And since Vicky did such a great job in introducing me, now you all know me. So I thought I'll start a bit of where we come from. If I can get the slides changing. Okay, here we go. So Jyväskylä is a city located at the central Finland, according to the population, not necessarily with the size of the country. And it's the 7th largest city in Finland with a bit over 140 inhabitants. So I would say roughly the same size as Colombia we're talking about here. And since Finland is the land of 1,000 lakes, we have always a bit of water surface area, even in the cities, as you can see in this picture that is from Jyväskylä. And even though we're not located at the capital area, it's about 3-hour drive to the south. Helsinki area. We try to be as international as we can, and currently we have a bit over 4,000 foreign residents from over 120 countries. And then I wanted to include some random facts here. So the works of the most famous Finnish architect, Alvar Aalto, some of you might have heard, can be seen throughout the city. And actually our faculty main building is also designed by Alvar Aalto. And the city hosts the Neste Oil Rally Finland, if you're into rally. This is part of the World Rally Championship, and this happens every year. And if you want to hear something about the behind scenes stuff, Enni's husband actually works as a reporter for this event. So please come to her and ask more about this event. And then maybe some of you have heard a Finnish comedian, a stand-up comedian, Ismo Leikola, who is from Jyväskylä, has been named as the winner of the funniest person in the world competition. And I just want to say he has lived a couple of years in the States. So if you Google him and find these clips, so there's a lot of things about the differences between Finnish culture and US culture and the language. So please watch those. You will understand the problems we have with the pronunciation and stuff. So yeah, I'll recommend that. Then there's some key figures about our university, just for you to get an idea of the size of the university. So we have a bit over 13,000 students with a bit less than 3,000 staff members. And this is the funding that we get from the government yearly. And of course, we seek for supplementary funding. And Academy of Finland is probably the biggest funding source that we apply funding from. And as you can see, we're working on the open access numbers. Of course, it would be great if all the publications would be available for everybody, but we're getting into the right direction, I think. Currently, we have 7 centers of excellence in research and 2 academic professors, which is basically the highest position that one can get in an academic career in Finland. And about the faculties, I'm sorry, I didn't have time to, I understood that the faculty has a different meaning in here than it has in Finland. So I would change this to department or a school. So 6 different schools. And me and Enni, we both work at the sport and health sciences. And again, replace the faculty with school. So our university is turning 160 years this year, and actually our school is turning 60 years. So it's a big year of celebration for us. And our school has about 200 employees, so it's a rather small unit, and annually we have 180 new master level graduates. And here's the list of the teaching subjects. So basically we go all the way from social sciences of sport, from the whole body level into the molecular level of exercise responses. And Enni and I both have graduated from exercise physiology. And since our school also has the gerontology research center, so we also teach subjects related to that. And since some of you may be teaching or looking for positions to study, so I just wanted to let you know that we have also 3 international master's degree programs. We have the responsible management and business of sport. We have the biology of physical activity, which includes also the exercise physiology, and then psychology of physical activity, health and well-being. So if you have anyone in mind, please recommend our university. This was my short introduction of Jyväskylä and our school. And do you have any questions at this point? You can come later on also and discuss with me. If there's no questions, I'll move on to my actual topic. So for this talk, it's based on these 3 articles, and I'm the first author of the first one and the third one, and the second one was authored by Maria Torres. And I had very minor role in there, but I thought it fits well with the story. So I wanted to include that in here as well. So my postdoctoral project concentrated on the effects of estrogen on small non-coding RNA signaling after an acute bout of exercise. And today I'm going to concentrate mainly on microRNAs as small RNA species and extracellular vesicles as a transport vehicle for them. But let's start with the very key topic for our talks, which is menopause. So menopause is much more than just hot flashes. It changes several aspects of women's health. Most of the changes are either caused by or at least associated with estrogen deficiency. And estrogen deficiency leads to, for example, elevated blood lipids that in turn lead to increased cardiovascular disease risk. So estrogen has health benefits, and so does exercise. So why not combat the unfavorable changes that are caused by menopause with exercise? Unfortunately, we have shown that even relatively high vigorous leisure time physical activity level does not entirely offset the lipid profile changes that are associated with menopause. Now, this led to the thought that maybe estrogen deficiency can also change the acute exercise response. To study this, we needed two groups of women, and we got these women from Eija Laakkonen's ESMIR study. For this study, we had altogether 14 postmenopausal women, of which 7 used estrogen-based hormonal therapy and 7 were non-users. And these women were matched for age, peak aerobic capacity, body mass index, and body fat percentage. In order to study the estrogen status and acute exercise on systemic signaling, these women went through a maximal test on a bike ergometer. And we harvested blood plasma samples before, immediately after, and 1 hour after the exercise. From the plasma, we isolated EVs, and we did this because extracellular vesicles are thought to be this active, targeted way of cell communication. So they don't carry just trash that the cells try to get rid of, but it's very targeted signaling. And in order to do this, we needed to overcome some issues that normally are related to isolation of EVs. So most of the time, if the sample originates from blood, the problem is that EVs are co-isolated with HDL particles. And this happens, for example, if you only use the density gradient ultracentrifugation method. And I had a great discussion about this with Jennifer today, and I could, use the whole talk time for this, but I'm trying to be brief. But yeah, so they share the same density. And it's important to consider that the EVs and HDL particles are normally together in the prep, since, for example, HDL particles carry small RNA species, so this may actually affect your results. So how to overcome this? So what we did, we used density gradient ultracentrifugation, and then we combined that with size exclusion chromatography, since luckily HDL particles and EVs differ in size. So by doing this, we could get pretty clear, nice preps of HDL and the other prep containing the EVs. From the isolated extracellular vesicles, we studied the small non-coding RNA species that are known to coordinate several physiological processes. So we did a sequencing. First, we took a look of these study groups together. And what we found out was that the relative abundancy of microRNAs and small RNAs that originated from transfer RNA increased, whereas small RNAs of ribosomal origin decreased in response to acute exercise. So when we compared the post time point to the pre time point. However, when looking at the top 100 small RNA sequences, the microRNAs were the most abundant at each time point. So hence we continued our analysis with the microRNAs. MicroRNAs are well known to coordinate gene expression, Typically, they work in the cell cytoplasm by binding to its target messenger RNA. And since microRNAs are short, they're about 20 nucleotides long, each microRNA has several target messenger RNAs. So that is, they are not gene specific. Once the microRNA finds a target messenger RNA, it will bind to it and the protein assembly is blocked. And hence the expression of that protein is suppressed. It is also known that the expression of several microRNAs change in response to exercise. And here you can see a figure of the most commonly altered microRNAs that respond to acute and or chronic exercise. And nowadays it's known that microRNAs are not only biomarkers of exercise, but that they actually also facilitate the adaptations to exercise. So in that sense, I would say that it's important to have the response in the microRNAs in order to get the adaptations to exercise. Now going back to our study. So when we took a look at the hormone therapy users and non-users separately, we found something interesting. So only the hormone therapy users had a response in the microRNAs to acute exercise, and there were no changes observed in the non-users. Next, we wanted to take a look at the possible function of these estrogen and exercise responsive microRNAs, and we did this by using two different online databases. And both of these databases pointed out that these microRNAs coordinate, for example, for FOXO protein signaling pathway. From previous literature, we know that FOXO is known to play a significant role in regulating whole body energy metabolism. And interestingly, in rodents and cell models, FOXO acts as a switch from the use of carbohydrates to the use of lipids as the major fuel substrate in skeletal muscle. And of course, when we talk about energy metabolism, we talk about mitochondria. And FOXO also acts in crosstalk between the mitochondria and the nucleus. Speaking of mitochondria, we have previously shown that estrogen localizes into the mitochondrial membrane. And here you can see the explanations of the groups. So we have normally cycling females. This was done in mice. And then OVX means ovarectomy, so removal of the ovaries, which will cause basically menopause to the animals. And it doesn't only localize into mitochondrial membrane, but it actually improves mitochondrial function. And as you can see in this figure here, it didn't affect that much of the mitochondrial complexes 2 and 4, but when we look at complexes 1 and 3, there's a clear restoration of the mitochondrial or the mitochondrial function in those complexes when estradiol is present. So what we're stating here is that estrogen may have direct effects on mitochondrial function, and then also microRNAs may work as a link between the estrogen status and the mitochondrial function. Furthermore, I have studied how estrogen affects microRNA signaling in skeletal muscle. And since estrogen deficiency is linked to diminished muscle mass, apoptosis could be one mechanism behind that. So we took a look both of the extrinsic pathway that comes the signal outside of the cell, and then also the extrinsic and then the intrinsic pathway that starts with the mitochondria releasing the cytochrome C. So these are the main pathways that can induce apoptosis. And what we found was that when there was the estrogen deficiency, many microRNAs that are linked to apoptosis were downregulated. And not only did we see the difference in the microRNA level, we also saw that some of the target proteins that these microRNAs have that are on apoptotic pathways, the messenger RNA level gets up, and also the expression of the protein is higher. So this may be one mechanism that could contribute to loss of muscle mass that is linked to estrogen deficiency. So to wrap up, estrogen deficiency caused by menopause diminishes the acute exercise-induced microRNA response in extracellular vesicles. And actually, we saw the same phenomenon also in HDL particles. What we suggest is that estrogen-responsive microRNAs may participate in coordinating the metabolism in response to an acute bout of exercise. And we also speculate that it may contribute to fuel selection, potentially by affecting the mitochondrial function in postmenopausal women. And of course, this is speculation. So now we are doing the same with the animal model so that we could actually get into the tissue level and to see how this affects. And what is exciting is that this is unpublished data from my rat study. So also in rats, the estrogen deficiency blunts the exercise-induced microRNA response. So with this setup, we can go into the target proteins of these responsive microRNAs and see whether they actually had some kind of effect, for example, in liver or skeletal muscle. And that's what I'm working on right now. And also we conclude that estrogens improve mitochondrial function. This can happen directly or possibly via microRNAs. And that loss of estrogens led to downregulation of the microRNAs and hence upregulation of the proteins linked to apoptotic pathways. And hence estrogens may contribute to the loss of skeletal muscle mass through the microRNA signaling. Then something about the future. So as Vicky already mentioned, I work as a postdoc in this ESMIR study that is led by Professor Eija Laakkonen. This already ended last year. And now, by the end of this month, I will end my postdoctoral project, this is kind of a funding source for me to become an independent researcher. And this is also why we wanted to have our visit now, so I could get it before this grant ends. And yes, we were aware of the weather things. So Vicky did warn us, don't worry. But this is wrapping up now, this project. And starting off 1st of September, I will start EVA study. So both of these, actually all of these are funded by the Academy of Finland. So I got a 4-year project to start my own research group. I'm actually hiring a PhD student right now. So if you know anyone who's available, who would like to study exercise, extracellular vesicles, and senescence, just please let me know. And of course, there's many people to thank from my own university, Eija Laakkonen, whose group I was part of. And now I'm hiring a PhD together with the head of laboratory, Maarit Lehti. And of course, a bunch of other people that helped me on the way. And of course, I thank people from the University of Minnesota, where I spent my postdoctoral year at the group of Professor Dawn Lau and Maria Torres from East Carolina University. Thank you so much for your attention. I think that would work, so I get the whole thing.
Okay, so I will then continue. So thank you, Vicky, for the introduction, and thank you for having us here. And it has been such a pleasure to meet all of you. We really enjoyed our time here so far. So I'm going to talk to you about eating behavior and physical activity, not so much about physical activity, but something about it as well for a healthy body composition during menopause. And Vicky told all of this already, so I can just skip it. I thought that I would shortly present these ERMA and ESMIR's study projects. So I'm going to present today results from this ERMA study. And the current PI there is Eija Laakkonen. So all this data has already been collected, and now we are just doing some more analysis. But this is a research project that has aimed to understand the molecular mechanisms behind the physiological level changes that coincidence with menopause. And this project collected a population sample of women between 47 and 55 years old. So we had over 1,000 women participating in this study. And then we followed some of these perimenopausal women until they became postmenopausal. So ERMA has been investigating body and muscle composition, muscle performance, physical activity, cognitive abilities, mental well-being, biological health metrics. We have tons of data. And here you can see the flow chart, how the study sample was formed. So I'm going to present the results from this cross-sectional part of the study, but as you can see here, and as I already said, some of those perimenopausal women were going through some follow-up measurements. And after that, we had this ESMIR study that Sira already mentioned. So it was a follow-up study to this ERMA project. So Eija Laakkonen is the principal investigator here as well, as Sira said. And we did the follow-up data collection for the ERMA participants when it was 3 to 4 years since their initial baseline measurement. So we repeated all the ERMA baseline data collection for 304 women. Unfortunately, the COVID interrupted this data collection a bit. So there's questionnaire data from almost 500 women. And this study investigates if and how women's metabolic health changes due to menopause. And in addition to all those things that had been done during the ERMA project, there's some new things such as 4-day food diary, eating behavior, and eating disorder questionnaires that we collected at this point as well. And then for the small part of the women, also indirect calorimetry at rest and during cycle ergometry test was done, and then also glucose tolerance test. Some of the women took part for that as well. And as you heard, this included also some experimental animal studies. So why did I get interested in menopause? I will give a short background of that. So as you might know, menopausal loss of ovarian function leads to a ceasing of cyclic secretion of estradiol and other sex hormones as well. And when these changes in hormonal levels happen, women experience a rapid increase in metabolic and cardiovascular disease risk. That's quite clear. And it has been thought that the cessation of estradiol secretion is the triggering factor for accumulating adiposity that then could also affect the increased cardiovascular disease risk. And it seems that in particular, the body fat distribution changes and the amount of visceral fat increases due to menopause. So it has been thought that perhaps the increase in fat mass is due to aging, but the changes in body fat distribution might be related to menopause. And as you might also know, overweight and obesity are major health concerns at all points across the lifespan, of course. And it is well known that dietary habits are related to the growing problem of obesity. So perhaps the increased risk of obesity may be influenced by inappropriate diet or sedentariness also during the menopause, or then perhaps it is the detrimental combination of these both factors. But actually, it is not known whether eating behavior changes over the menopausal transition and what is its role in the changing body composition. And that is something I wanted to look at. So now I have this, as Vicky said, this own sub-study that utilizes the data from ERMA and ESMIR's projects, and it has quite a long title, but the acronym is HEATMENT, and I think the environment is now very good to talk about HEATMENT study here. So the aim of this study is to examine the role of eating behavior and physical activity in sustaining healthy body composition during menopause. And then we are also trying to look how mental well-being and menopausal symptoms associate with the mentioned health behaviors, and then what is their role in sustaining physical health and healthy body composition during the menopausal transition. So there are many things involved, as you can see from the picture. And now I'm preparing my first manuscript from this data. That is, and its title goes Visceral Fat and Eating Behavior Across Stages of Menopause. So an individual gains weight through positive energy balance, that is, energy intake is higher than energy expenditure. And the type of amount and type of food we eat has, of course, an impact on energy intake. And before we know how much an individual is eating, we need to take into account the eating behaviors that include the choices about when, where, what, and how much to eat. And it is known that sex hormones play essential roles in the regulation of appetite, eating behavior, and energy metabolism, and that food intake and reproductive functions are linked somehow, because appetite and food intake may vary even during menstrual cycle-related hormonal fluctuations. And I think it's quite clear from the animal studies that loss of estrogens increases food intake, but it is not known whether this is the case also with humans. So there are not so many longitudinal studies where the menopausal women have been followed from premenopausal stage to postmenopause. But there are a few studies, and one of them showed that there were no changes in total energy intake or individual macro or micronutrient intakes among women over menopausal transition in comparison to women who stayed premenopausal during a 5-year follow-up period. And then there's also some evidence that menopausal transition is associated with increased appetite. Then on the other hand, it might be associated with increased restraint in eating. And also it seems, or it might be, that prevalence of disordered eating or eating disorders and body image concerns are increased during the menopausal transition. So the purpose of this observational cohort study was to investigate whether there are differences in eating behavior between pre, peri, and postmenopausal Finnish women, and whether eating behavior and food intake associate with the amount of visceral fat. So this is the cross-sectional data of the ERMA study, and participants were 47 to 55-year-old women, so approximately around 50-year-old, and they were assigned into these 4 groups, so premenopause, early perimenopause, late perimenopause, and postmenopause, according to their blood follicle stimulating hormone levels and bleeding patterns. And these women were quite healthy, so they had their BMI around 25, their body fat percentage around 30, and they were moderately physically active women. And we examined their eating behavior with a structured questionnaire. There were 13 questions regarding different eating styles. You will see those questions a couple of slides later. And then we investigated their food intake with a 45-item food frequency questionnaire. The visceral fat was assessed by a bioimpedance device, so in-body machine. And then we had the physical activity. It was self-reported leisure time physical activity question with a 7-point scale from sedentariness to competitive training and several times per week. So we tested eating behavior, food intake, and menopausal status for association with visceral fat by linear regression analysis. And here you can see the confounding factors. So we put the self-reported physical activity there, and it was recoded either as low or high. And then we had the depressive symptoms measured by CESD scale. Then we also had age, body height, and educational status. And then we also did some analysis with ANOVA, and it seems that in our data, there are no differences between the menopausal groups in the amount of visceral fat, eating behavior, and food intake, or in the level of leisure time physical activity. But as you can see, all the confounding factors were significantly associated with visceral fat. And luckily, logically, the more the higher your physical activity level is, then it is associated with lower amount of visceral fat, for example. But after adding all these confounding factors, we also got some statistically significant associations with these eating behavior items, and then also the food intake. So here you can see all the eating behavior items that were included in the regression model. So we asked about their snacking styles, whether they are eating or having highest food consumption in the evening, whether they are attempting to maintain healthy eating patterns, whether they are avoiding fatty foods, and so on. And then we also asked about the restraint in eating, so how much they try to restrain their own eating, or are they eating whatever and whenever they want. And I will go through these with these different tables. So here are all the eating styles that were statistically significantly associated with the amount of visceral fat, and an attempt to maintain healthy eating patterns was negatively or inversely associated with the amount of visceral fat, and then all these other items were associated with higher amount of visceral fat. So snacking type of eating, I would say evening oriented eating, some aspects of emotional eating, and then also restricted eating were associated with higher amount of visceral fat. And the only eating style item that had some interaction with menopausal status in the regression analysis was this grazing throughout the evening. So otherwise the menopausal status didn't play any role here. Here you can see the food items. So we asked how frequently they consume these certain food items, and here are some of them that had a statistically significant association with visceral fat. So in those first three rows, you see that the association with visceral fat was not significant, but there were some interactions between menopausal status and physical activity level. But dressings or oils with vegetables, sausages, cold cuts, and hamburgers, the higher the consumption of these food items was, the higher was also the amount of visceral fat. And then there are still few, so candies, low calorie soft drinks, functional food items that mean like, for example, juices that had been added, where some vitamins added or probiotics added, they were positively associated with visceral fat, and then nuts and seeds were negatively or inversely associated with the amount of visceral fat. So it seems that in our cohort, the menopausal status is not that important, but there are some things that in your eating behavior and food intake that might affect your visceral fat. And then I thought I would say a couple of words about sleeping, because I have now one PhD student. She started doing her work and she got interested in the sleep part. So we are now trying to find out whether eating and sleeping behaviors interact during menopause. So Hanna-Mari Lankila is doing her dissertation at the moment, and she's working with her first article, or actually it has submitted already. So it is known that eating and sleeping behavior interact with each other, but the research is limited in the context of menopausal women. It is known that 40 to 60% of menopausal women reports sleeping problems, such as insufficient amount of sleep at night, reduced perceived sleep quality, or feeling daytime tiredness. Of course, there are also studies that show that you sleep completely well, even though you are going through menopause, so the evidence is not, or there are evidence from both sides. It has also been shown that menopausal symptoms may contribute to poor sleep. And the aim of Hanna-Mari's study is now to examine whether menopausal status is associated with perceived problems in sleeping. And we also studied different aspects of eating behavior as potential risk factors for poor sleep in menopausal women. So over 20% of the participants in our ERMA study reported fairly poor or poor perceived sleep quality. So the amount of sleep problems seems to be a bit lower in our cohort than it is according to literature. But the amount of women who slept less than 7 hours per night was 18% in premenopausal women, 17% in early perimenopausal women, 20% in late perimenopausal women, and 25% in postmenopausal women. So it seems that the amount of sleep you get is a bit shorter when you are postmenopausal. But in this data, the menopausal status was still not associated with any measures of sleep in regression models. But what is interesting is that women who reported more snacking type eating behavior, they were more likely to report also lower perceived sleep quality, shorter sleep duration, and more daytime tiredness. And also externally cued eating, so eating, or it means that when you, for example, seafood or seafood advertisements you want to eat or eat after that, that was associated with shorter sleep duration, and emotional eating was also associated with experiencing daytime tiredness. And it is known that sleep duration may be negatively associated with the amount of visceral fat mass accumulation during adulthood. And I think that is why these connections between eating behavior and sleeping problems is also interesting in the context of menopause. So my conclusions so far would be that in particular, more frequent snacking and evening-oriented eating, I'm not sure if that sounds reasonable term to use to you. You can correct me if you want. Then some aspects of emotional eating and stronger desire to restrict one's food intake. All these were associated with higher amount of visceral fat. So our results suggest that to hamper the increase of visceral fat that takes place during menopause, it might be important to pay attention to eating behavior and not only the foodstuffs we are consuming. And it also seems that eating and sleeping behaviors are linked during menopause. So women who experience sleeping problems might benefit monitoring eating behavior during menopause and vice versa. So if we are monitoring the eating behavior of those women, perhaps we should also then check how they are sleeping. And of course, in addition, it is important to stay physically active throughout the menopausal transition. And I would add that it's also important to take care of your mental well-being, but that's perhaps another story. As you can see, we've had some time to explore the surroundings. It's been very nice. Thank you for your attention, and I believe now we are ready to take your questions. Yeah, good question. Yeah, well, I have to say first of all that from this we don't know what they were actually eating, so we don't know what they were snacking or how they were what they were craving throughout the evening. We've been talking about the snacks in here and in Finland, so it might be a bit different. But of course, we do have all those chips and cookies and what else. And I assume that if you are snacking more in the evenings, then it might be that those snacks aren't the most healthy things. So perhaps the evening snacking goes along with the more fatty and salty things to eat. I'm not sure if I'm answering your question, but this kind of speculation I could give.
I think they had several different types. Product can be variety. I'm gonna have to check up. All the eating. I'd say 13, 19.
Yeah, actually, I didn't put that reference here. So these are from it's a Finnish researcher, Keski Rahkonen, who has developed this, and it has been validated in, I would say, in obese adults. But yeah, it doesn't have any like title or name, but I can pass it to you. What? Yeah, actually, we have that from the ESMIRS, so from the follow-up study, we have that. Yeah.
About 30%.
I think it's more. I think it's around. I would say that overweight is around 40%. Could it be? I don't remember. We have to check.
I'm sorry.
Yeah.
That's okay. I was expecting it to be less.
In the animal studies where we did see, the dramatic difference in the fat accumulation. Is basically, I think you can harvest like all of it, kind of comparable amount. And then for the muscles, I only have the lower limb muscle. That's basically.
Yeah, I think the data shows that the lean mass goes down with the menopause and visceral fat goes up. Well, I don't think we have checked that like that. Yeah.
Cohort,
yeah.
Probably, but in.
Yeah.
Being done in our school, but before we joined the group.
But also the physical activity seems to counteract some of the effects of loss of estrogen. Yeah,
recall?
I think so. I don't.
Set of data,
Yeah.
so sorry, we're kind of like here trying to cover every aspect, but
Yeah.
I think those were reported in one of the publications, but I don't remember by
Yeah, me
heart
neither.
the.
Yeah, and then now some people have done these arterial stiffness things,
Oh,
so they
yes.
have combined this ERMA cohort to another cohort. So Eija has looked at that as well.
Oh, you mean the humans? This is. And so-called. So they were pretty. Higher intensity stuff. Our understanding, there was no decrease. All these women say of all of these things happen with the menopausal phase. So, but the signaling thing that I'm studying, I hope it's an acute response. Of course, the microRNA signaling and extra. At the time points, like before the exercise and then immediately. Actually, this project that will end now was concentrated. So for the next one, I kind of dropped it out. So in that sense, I'm not going to go like that much into that topic. So I'm kind of hanging on with the extracellular side. But it's a good question. And of course, with the microRNA side, I'm going to take a look at what's inside the vesicles and then. Of course, the functionality of the microRNA. Thank you very much. Thank you.